Okay, welcome everybody. We'll just give it a minute to let some others join. So yeah, one more minute and then we'll start the meeting. Okay, I'm sure others will join, but we'll maybe make a start. Welcome to this webinar. My name is Jan Turner. I'm from Humane World for Animals, one of the founding members of AFSA Collaboration. And today we have a speaker from PathoQuest, Colette Cote, who will speak to us about NGS. I just want to share my slides. Wait a second. So hopefully you can see my slides. Yeah, great. All right. So today's webinar will be, as I mentioned, from PathoQuest, one of our AFSA members, and it is on biopharmaceuticals animal-free viral assessment. So in terms of just some housekeeping rules today, all participants will be muted and their video will be turned off. If you do wish to ask us anything, you can please use the chat function in Teams at the top. Or if you have a question, please do feel free to add questions to the Q&A function. And we'll go through the questions at the end of the session. Oh, sorry. So in terms of this webinar, this is another event in our Humane World for Animals project, accelerating non-animal batch release testing for human vaccines. This project has been going on for nearly 3 years, and it allows stakeholders to have dedicated discussions with our experts in the field to fill in any knowledge gaps and allow additional discussions on prioritization of non-animal testing strategies for batch release testing. You can go to the link below, which gives you all the information on this project. The objectives of the webinar today are really to engage industry and regulatory stakeholders in the application of next generation sequencing for biosafety testing. NGS has really reshaped the way biologicals are tested for adventitious agents, offering an agnostic animal-free approach that accelerates timelines and improves the safety of biologics. With the recent updates to the global regulatory guidance documents endorsing the use, NGS is now well positioned to be a critical and central component to any biosafety testing strategy. So our speaker today, as I mentioned, is Colette Cote, General Manager and Chief Scientific and Portfolio Officer at PathoQuest, and she will provide you with some essential details and case studies on how to implement, simplify, improve and expand the adoption of NGS-based biosafety solutions for your own pipelines. This talk will provide you with an insight into current regulatory expectations for NGS applications, specifically what the regulators are asking for and why. The webinar will last for approximately 45 minutes, and then we'll have around 10 minutes of questions afterwards. And the recording will be made available on the AFSA website, which is below at the bottom there. And we last month had a workshop describing the use of NGS in replacing the monkey neurovirulence test, the challenges and opportunities for the future. So that's enough for me for now. I will, apologies. I will now hand over to Colette to start the webinar. Thank you, Colette.
Thank you. Thank you, Jan, and welcome everyone. Hope everyone having a great day so far. So I will now share my slides and hopefully those are visible now. Yes. To the team here. Perfect. Okay. So again, thanks for the opportunity to give a little bit more insight about the use of next generation sequencing to replace animal-based testing for biopharmaceutical safety testing. So I'll give a bit of an overview of where we are in the field, and a little overview again on next-gen sequencing for those who might be less familiar with it, and then go through some specific applications and some data that we have in terms of acceptance of that, and then add a little bit more in terms of the regulatory aspects of it. So first here, I'll just go through, I mean, this audience is probably pretty familiar with the entire biologics manufacturing process, but I just wanted to highlight a few things here. This is a high-level overview from a company like PathoQuest, which are a small CRO. We deal with materials throughout, or different modalities throughout the manufacturing and production life cycle, whether it's viral vaccines or oncolytic viruses, gene therapy vectors, traditional mAbs, and so forth, cell therapies. A lot of different materials need testing at various points. Typically, that testing falls into either adventitious agent detection, which is your virus hunting testing, or genetic characterization. One of the key points that I just wanted to highlight here on this slide is during that manufacturing process, your raw materials will likely go from a cell-based system to a cell-free system. There's a transition point typically in which your product is then purified from the sample and essentially vialed for use with a patient. Throughout this entire scheme here, next generation sequencing can be used. But before I get to that, let me just talk a little bit about traditional biosafety testing. So the way that it stands right now, a lot of these tests are decades old. And the specific test package that's required for your modality will depend on exactly what that product is, as well as the regulatory guidance that is surrounding it. What these are here in this middle panel here, this is a collection of multiple aging assays that run the gamut from animal-based to true molecular-based testing. For the animal-based testing, you have your in vivo assays, and then you have other species-specific assays, including HAP-MAP-RAP assays. Those are quasi-animal-based assays that again have long been in the field. However, this collection of tests is traditionally considered a low resolution test. Low resolution, they may have issues with specificity. They may lack a susceptibility to certain viruses if you're looking at virus detection. There can be extended timelines as it applies to animals or culturing-based systems. And sometimes there's an increased cost based on that, just depending on if things need to be repeated or even the length of time that these assays are being run for. So if you look at these assays as a whole, you could say, what are my classical test concerns? As I mentioned, it's resolution. If you're looking at an animal-based test or in vitro cell-based test, you see a cell may have a problem, CPE, the animal might be struggling health-wise. You don't know exactly what the cause of that is. Is it directly related to your product or not? You don't know. And it take a number of orthogonal approaches in order to really identify what's going on there. Again, as I mentioned, specificity, not every test system is designed for every imaginable viral contaminant that could be out there, particularly as the new ones emerge. For a while, Zika was a bit, concern, obviously, coronavirus and so forth. As I mentioned, there's a lack of test system susceptibility, and there can also be assay interference. So in some modalities, oncolytic viruses, they're going to destroy your test system if it's a cell-based test system, just by the nature of the product. And there's also a requirement to understand if there is assay interference or matrix effects going on with your test sample. So this is a large collection of assays. And when you're working in the world of traditional tests, the goal, of course, is to try to have them more ethical, to reduce the number of tests, simplify them, and so forth. So I just wanted to highlight some of the features of obviously animal-based testing. These are 50-plus-year-old assays. They may predate a number of us in this on this webinar today. On the left-hand side, it just goes through some of the features of these assays. So from an in vivo assay point of view, you can use variable numbers of animals, whether they're adult or suckling mice or embryonated chicken eggs or even guinea pigs. And there tends to be a pretty high false positive rate and false negative rate compared to in vitro assays. And this was actually a publication that came out in 2022 that did an industry-wide evaluation of this. Likewise, HAP-MAP-RAP assays, these are quasi-animal assays, so they have an animal starting point, even though they have a immunological endpoint. They do require use of animals as well, and they have higher repeat testing rates. So these are less ideal assays, and the newer guidance documents have really shied away from these assays and said turn to additional or newer assays that eliminate the need for animal-based testing. Obviously, these assays are time-consuming. Again, poor resolution, poor specificity, poor sensitivity, and then you can't always identify what is causing the problem. So our goal, at least my interest, of course, is also to move these assays away from animal-based testing to molecular-based testing, particular next-generation sequencing. So if you look at a simplified biosafety testing approach, again, the goal, can we develop one assay or one test that combines or replaces several assays specifically for the animal and quasi-animal based testing? And again, the guidance documents already very much support the 3Rs principles and to eliminate animal-based testing, but also looking at other things like PCR-based tests, the in vitro cell-based tests, immunoassays, the 9CFR tests, and potentially some others. Now, I don't have just NGS here by itself. There are some tests right now that don't quite fit into an NGS workflow. However, we're looking into these and we're working on them. For example, mycoplasma is something that can be done, but there are some considerations that are needed. Same thing with retroviruses and latent viruses. There are some considerations here. So we still leave these a little bit aside from NGS, but NGS essentially will replace, or the goal is to replace all of the other orange cogs that had been seen here. So the advantages, and I'll go through these as I go through this talk, is improved resolution, specificity, and sensitivity, simplifying matrix interference assessments and method adoption, because that's a key consideration. And then in some instances, depending on what modality you have, there's we lessen the need or eliminate the need for neutralizing antibodies or other pre-work, including manufacturing of control cells and so forth. But before I get there, let me just go through an interesting history of sequencing for those who may not be as familiar with this. The era of next generation sequencing or high throughput sequencing, massively parallel sequencing, it's had a few different names over the years, has really been at the early 2000s. Prior to that, you're familiar with Sanger-based sequencing in the mid 1970s all the way through. But really the transition point into the second generation of sequencing happened in the early 2000s. And in fact, in 2008 is when next generation sequencing actually entered the biosafety testing space. It was only in about 2014, 12 to 14, where GMP level NGS assays were developed and launched. But there's been a lot of platform changes throughout this period of time. And of course, with every platform change, the goal is to make it less expensive, faster, more data, less expensive, and faster to meet the needs. So this is very much an evolving area. Sequencing is. Again, with the different platforms. What it is today, though, is NGS is a 20-year-old technology now, almost 25-year-old technology that is very explicitly stated and included in international regulatory guidance documents. And it has been included into regulatory filings. So it's getting a lot of visibility from the different health authorities and regulatory agencies globally. And we can see that in different areas. We can gauge that, in fact, through certain things like a Type 5 master file. And I can talk a little bit about that. So this is just a very interesting historical scheme. Where we are at PathoQuest, we use two different platforms. We use the Illumina system as well as long-read sequencing using Oxford Nanopore systems. But there are other systems out there that can also be applied for these specific applications. But what's the difference between these sequencing systems? And again, this might be information you already know, but we'll just clarify it here. First generation is really your Sanger-based sequencing. So this is sequencing by synthesis. This is typically considered shorter read sequencing, typically less than 1 kb. It is primer-based. So you essentially have to walk across a known sequence using a known primer-driven sequencing reaction, and it's consensus-based sequencing. So you're taking the consensus base call and not individual base calls. And this is still used today. It's a very robust sequencing method, and it has its applications. But the newer generations of sequencing are the second and third gen sequencings, and they differ a little bit from each other, but they differ significantly from Sanger. So so a second generation sequencing system, like a Roche system, or the early days of the Roche systems, are ABI and some Illumina systems now. What this is it's still considered short read sequencing. So this is less than 300 bases currently is the max that we get per sequencing reaction, but it's also sequencing by synthesis, and it generates millions to billions of data sets. So an individual, I should say, reads within a data set. The third generation is more associated with Oxford Nanopore technologies and the PacBio systems. These are typically longer reads or all read sequencing. I know some of these vendors prefer to call themselves all reads, but they really are geared towards longer read sequencing, and some can reach upwards of 200 kb in length. These data sets don't tend to be as large as Illumina-based data sets, but they have their specific applications in the field. The difference between first gen and these later generation sequencings is these later generations are sequencing agnostic. In other words, you don't need to know anything about that sequence that you want to identify because you use adapters that you tag the ends of sequences, these molecules with, in order to sequence them. And they're also significantly higher throughput. Here, again, we're talking billions to billions, and some of the biggest instruments, you can run 400 transcriptomes, you can run, a massive number of genomes in a single sequencing run, which is often an overnight run. So that's a little bit of the history of the sequencing systems here, but just to go into the basic steps, because they're pretty simple across the board, are pretty standardized. First step is, of course, sample processing. So this is just nucleic acid extraction and a process called library preparation. The platforms differ a little bit as to what you need to do here, but essentially you want to extract RNA or DNA or both. For some systems, you need to convert that to double-stranded DNA and size it appropriately so it can be sequenced. That's more on the Illumina-based systems. And you put these adapters on the ends that can be used for barcoding purposes, so you can multiplex and sample identification. And the sequencing primers, even long-read sequencing can have similar processing. These then get sequenced on your platform of interest. After that, you have a data set, a significantly sized data set that will then go through some level of data analytics and bioinformatics. And typically that includes things like data integrity checks. You have to move the data into an environment to do the processing. Any pre-processing to remove bad data, so remove poor quality reads, trim the ends off the adapters. They're not the most meaningful information after sequencing. Then your specific data processing, and then, of course, considerations for data archiving and storage. The considerations here, just to snapshot this, is really quality in equals quality out. So you want to have good material in, and you want to gauge that material and assess it throughout the process so that you know that your data set is a high quality for the analysis. In our world, these have to be GMP workflows, and even including the data analysis part of it. So for the bioinformatics, typically now most entities are using cloud-based computing because of the scale that we need for this. For data processing, data storage, data transfer. We also need to make these scalable environments because data sizes can get, they can get exponentially larger. We do build our own pipelines in the field, and they also have to meet current regulatory guidance requirements and client expectations, but they also are expected to be standardized to some extent. And there's a lot of consortia out there for this specific application. And then, of course, with virus hunting, we also need to ensure that we don't try to increase the level of false positives, and we have some level of secondary qualification of the data in order to minimize that. So that's the sequencing steps in a nutshell. I'll just add one more element here. To make a simplified testing strategy, doesn't matter how you do the sequencing, but what really matters is what your sample looks like. And I mentioned earlier already that there's a transition point from when you go from a system that is cell-based to a non-cell-based system or cell-free system. And it does have a little bit of an impact on sequencing, particularly for virus hunting, because it does drive what you're looking for a little bit differently. So there's essentially a decision tree, and I've simplified this here. For next generation sequencing, if you have cells present and you're doing virus hunting, what you really want to do is shunt yourself into what's called a transcriptomic assay. This is a cell-based assay. You take advantage of RNAs being expressed by the cells. The assumption is if a virus is replicating or is active, it will go through an RNA phase. That RNA phase will be easy to detect with this specific assay. And then we can use certain features of that RNA to determine whether something appears to be replicating or not. So we can use strandedness, for example. This assay here really is the legacy assay in the field. It's been out for as long as I've been in the field, so almost 18 years. And again, it's designed for things like master cell banks, working endoproduction cells, Livka cells, crude bulk harvest, and so forth. And so I'll go through some of the features of this assay, a specific assay shortly. On the other side of that is if you have a cell-free system, we can't take advantage of the same considerations that we would for a cell-based system. But here, really, we're interested in viral particles, intact viral particles, to ensure that if there's, if a virus is replicating, it will be in particle form. So the test materials here typically are clarified bulk harvest, drug products, drug substances, even raw materials. We call this our viromic assay. Both of these assays have the same general features, which are to provide simplified 3Rs compliant testing strategies with rapid TATs, broad detection capabilities, can detect any kind of replicating virus with sensitivities that are comparable to PCR. So PCR is effectively our gold standard that we try to compare ourselves to, and as well as to publish head-to-head comparability data compared to classical testing, so we know exactly how well NGS is doing in the field. So let me just give you some high level of our transcriptomic assay. So this is the way that we actually conduct this assay here and some of the features of the assay, just to kind of position you for that. And then I'll go into some of the background of, some of the data that we're actually seeing for this. So this assay, as I mentioned, is cell-based. So your test sample is typically, again, master working cell banks and so forth. The assay-specific controls include positive controls and negative controls, and these are designed in a very specific way. Your negative controls are just cells, we use MERC5 cells in particular, that are free from viruses. So they've been characterized, they do not contain a replicating virus, and therefore should be negative through our assay. Positive control cells are cells that are latently infected with, or we use latently infected samples. In this case, we have two viruses of concern that we track through our positive control. And this positive control is actually a mixture of infected to uninfected cells. So essentially one infected cell mixed in 10,000 uninfected cells. And of those, if the sample, we can look for the presence of both Epstein-Barr virus, or HHV4, as well as murine leukemia virus, which are the two known contaminating viruses in this particular cellular mixture. In addition, we use these synthetic RNA controls. These are spiked into the test sample in positive control particular. These are completely synthetic, so they're unrelated to any viral sequences. They're of varying sizes, they're of varying concentrations, they are certifiable. So we spike them in at specific levels. There's 92 of them in particular. We look at subsets of these for different features, but essentially we want to track these RNAs, the presence of these RNAs, the detection of these throughout this process. And this is essentially to counteract matrix effects. So we look at recovery of these RNAs in the test sample versus the positive control. And if the recovery is similar, we say we're not seeing matrix effects. If the recovery is not, then we can say that there is a matrix effect. And this is really designed for method adoption to eliminate some requirements for method adoption. It's already built into the assay. Once we have these samples, the key here is to do RNA extraction and then convert it to a library, as I had just mentioned. So this is, again, for in Illumina system, it's converting it to double-stranded DNA of a specific size range. These have adapters on the ends. The adapters are the sequencing primers, in addition to barcoding, allow us to do sample-specific or multiplexing of the sample. Once we have the library, it goes into, we use a NextSeq system, so it's a higher format system. We can generate up to 400 million, almost a billion individual reads or data points. That data then has to go through a data analysis step. So this is through our own proprietary pipelines. We use cloud-based computing for this with validated pipelines to basically ask for virus hunting. Is it a virus? Yes or no? If it is, I'll keep it and I'll do additional qualification of that read to determine, is it still a virus if I compare it against everything in GenBank, for example. And then once we have the short list of the sequences that we see, this goes to one of our viral experts to try to put it into context of the test sample. So there's nothing worse than just getting a data dump on you. What you really need to do is to understand how it is how it applies to your test sample. For example, if we see an avian virus in a CHO cell line, it doesn't make any sense. It's likely coming through a reagent. However, if you see murine leukemia virus in a CHO sample, we would have to therefore try to identify what the probable source is for that. And at the end, provide an actionable report. Now, what we have, these green boxes are these in-process control points. These are just go, no-go gates to ensure that the material that comes out of one step is suitable to go into the next step. Excuse me, because we know that this is a long and can be expensive process at times. And we don't want to have failures at the back end when we can prevent a failure at the front end. If you compare this assay, so that's a pretty high level view of the assay, but if you compare it to existing or legacy assays out there, this assay can be done in 2 to 5 weeks, so it can be faster than our standard 28-day assay, certainly faster than in vivo assays. And at the end of it, you do have identifications of what the offending agent is. So it can detect the unknowns, whereas these other assays will say there's something wrong, but they can't tell you what it is. And then PCR, remember, PCR is targeted, so you have to know what you're looking for. We also use lower sample input requirements, so we preserve more sample for downstream uses. And I've already gone through specific controls for the assay, which then alleviate the need for method adoption. And then here we also use something called RNA strandedness to assess active replication. Remember, this is a molecular-based test. And so in order to look at biological application, we use the strandedness. So for example, if it's a virus with a known DNA genome, if you start seeing RNA sequences of it, it's replicating. If it's an RNA genome, if you see, for example, if it's a negative strand RNA virus and you see the positive strand, again, you're looking at replication there. And there's a bit of data out in the literature available now with head-to-head publications on it. We actually have one through our partner with Charles River. These are publicly available publications that you can look at to see how this assay in particular compares to both in vivo testing in particular. So just a snapshot of, we always like to show what the data is. So this is just a very small snapshot of some of the testing that we've done. So we looked at 119 total studies, and out of them, 15 had a positive hit. So that's about a almost 13% hit rate on there. Now, remember, I said we go through this qualification process to eliminate a lot of the false positives in there. What we see primarily after that process is we get some very specific hits, but this is across the board. So as these pie charts down here show, the different material types that we're testing, predominantly master cell banks, a little bit drug product, drug substance for different reasons. I know this is not the primary use of the assay, but really working cell bank and master cell banks. In different clinical phases, it's across the board. And very much different sample origins, although rodent and human tend to be the most prominent ones. And then the modalities that we typically see the testing for this is right now cell therapy, at least of this sampling size. And what we've seen is, murine leukemia virus, HHVs, these are typically, CHO-based, human cell-based. BPV3 is often coming from reagents. Rhabdovirus is typically from your SF9 cell line or insect cell lines. Pestivirus, from, again, some raw material based or FBS based sequences that we see as hits. So again, going through a pretty rigorous qualification process. And then after this, obviously with NGS, while we have identification, we can make recommendations to do orthogonal confirmation testing, but these typically are pretty sound testing. We're looking for the presence of open reading frames, sequence signatures that say that it's different maybe from a control that we use, or it's different from what you would expect in your cell line and so forth. So again, it's been used pretty well, and we do see positives that would come up that may not necessarily have been detected by other traditional means. Now, most of these are part of panels, but we can certainly detect those. And just one quick case study here for this. So I won't go through the assay portion of it. Again, there's some more details there, but this was a human master cell bank from a phase 3. It was a cell therapy product. We had over 150 million reads. We do have a cutoff of 65 million, so this is well above that. All of our positive controls passed. We detected a mammalian orthoreovirus that did look like it was presence of infectious particles. We saw reads with eight of the 10 genomic segments, so 21% sequence coverage, 97% sequence identity match to what we identified it as. And then through stranded analysis, we were detecting the sense transcript. So that gives us our complementary verification that we are detecting something real in this test sample. So again, that's just one example. What I want to do now is just switch gears to the cell-free system. Everything from library preparation downstream is the same as I've just said, so I won't go over that again. I'll just go over a couple of different features here. So this is geared towards, again, cell-free materials like virus stocks, virus seed stocks, masters working, bulk harvest, drug product, drug substance, so forth, supernatants. You could even use it to some extent for raw materials. You have your test sample. We include a positive control. So this is a mimic sample that contains representative RNA DNA viruses. These are viruses that we use to track, similar like we do with the positive control for the transcriptome assay. We can track the detection of the viruses that we spike in here, and these are well-characterized viruses throughout this process to see, what level we're detecting. Again, we don't use straight RNA detection for the sample because we're looking very specifically at viral particles and the content, nucleic acid content within those particles. So we use ultracentrifugation or centrifugation steps in order to capture those particles and then extract nucleic acids directly from those particles. We do a separate RNA and DNA preparation step here to enhance for the detection of those sequences, but then it goes onto our sequencing platform. We make those libraries, it goes onto our sequencing platform, and then we evaluate the data. When you compare it to, against traditional tests, all the same that I've mentioned applies here. This assay is also faster than standard 28-day assays, also uses less input, and it does have LODs that are comparable to PCR assays. Now, because of the different controls that we do here, method adoption isn't as straightforward, but we can do spiking studies to assess matrix effects. In other words, we can use the WHO collection of spikes, viral spikes, into a study like this to assess recovery of very specific viral sequences. If I go through the snapshot here again, so this is just a sampling of 63 total studies. 10 of them had a positive hit, so it's about 16% rate. What we see mostly are pestiviruses, alpha herpes virus, and this Copi parvovirus. These are typically related to the use of serum. And it's pretty standardized across the board. We often detect these sequences. From a material type, again, just to kind of give visibility to what's been tested and how, most of this has been master virus seeds, although you can see drug product is another significant material type. Some in phase 1, some in commercial phase testing, other is just kind of classifying everything else. Majority are human or non-human primate, although we do have tested bovine and rodent-based samples. And this has been primarily in the vaccine, for the vaccine modalities, although you can see it does range the gamut here as well. So very similar to transcriptome, just in terms of the profiles that we're seeing. I just wanted to highlight a case study for this one as well. So it was a phase 1 viral vaccine manufactured in a human cell line. We ran our GMP assay. We had a total of 150 million plus reads for the 2 arms of the assay. All controls passed our acceptance criteria, so we knew it was the performance of the test met specifications. In this particular sample, detected bovine alpha herpes virus type 5 and pestivirus A, or BVDV. Again, this was verified using what we call complementary bioinformatics analysis. So we dive deeper into what are we hitting, how are we hitting it, how much are we hitting, is it an open reading frame, whatever, is it a regulatory element, whatever. We know that one of the controls that we use does have a BVDV as a known contaminant in it. However, this BVDV detected contained a very different SNP pattern than what we know as our standard BVDV. So we have the ability to discriminate between closely related sequences to then confirm that this was a BVDV contamination that was not as a result of one of the controls that we've used here. So again, just to show the power of this, the ability to detect and even discriminate from some of the other features of the assay that we have. And I want to highlight this was actually a poxvirus master virus seed that was used as the test sample. So let me just switch gears here to some of the regulatory acceptance of this of NGS, and it's been a very interesting foray in this. This is just a brief timeline from 2010 up to current or present day. As you can see, there's been a lot of regulatory guidance documents that are very specific for the industry. Prior to the 2020s, I would say a lot of the language in these guidance documents, NGS would fall under the word state of the art. So state of the art technologies, you could group NGS. It wasn't explicitly spelled out. However, with some of the newer revisions, now NGS is explicitly spelled out as a technology to be used or to be considered, particularly in light of animal-based replacement for animal-based testing, particularly so with the ICH Q5A revision here. And then we have a lot of other documents in different fields for different modalities, but effectively, all of these guidance documents are pushing for use of NGS and other technologies like it for the replacement of animal-based testing, as well as the replacement of other testing. So, for example, to use the state of the art for testing strategies. If I dive a little bit deeper here from, again, regulatory, again, this is some of the language here that we're seeing. For example, ICHQ5A, which came out a year or so ago, recognizes NGS very specifically as an alternative to in vivo, in vitro, the PCR assays, 9CFR based testing, adventitious agent-based testing. There's, in fact, new European Pharmacopoeia chapters very specifically for the use of NGS and the validation of its NGS. What specific validation strategies should be taken, what considerations need to be made. And this is in addition to a number of other chapters here, 5.2.3, 2.6.16, and 5.2.14, which also call or endorses NGS for various testing strategies, but very specifically, this European Supplement 9.3 details the substitution of in vivo methods by in vitro methods for the quality control of vaccines. So again, collectively, these guidance documents very much support NGS. Even those from the WHO, the EMA, some of the newer guidance, particularly around ATMPs, and of course the FDA has a number of workshops that are going on, and there's a number of consortia as well that we also take part in to try to standardize NGS and make sure that the agencies really do understand what this assay is and how it can be used for this particular application. A couple other points here to mention before I wrap up. There are additional regulatory support and considerations that must be made. We are a small CRO that specializes in NGS, so our assays must always be built to meet global and changing, evolving, and sometimes conflicting regulatory expectations. And we can see that and we try to accommodate it. We also see that as modalities change, we need to keep pace with those and provide more tests or maybe advance our tests in order to continue to meet the current expectations. We've also taken the initiative to present our portfolio, very specifically our transcriptome assay, to various agency innovation centers. And we're talking the ANSM, EMA, the FDA, and other authorities globally, where we have dedicated sessions as part of their ITFs or their CAT committees. Again, these are the technology sides of the agencies, where we are able to present on the assay, present what it can do, even present sometimes what it can't do, present on what we would like it to do. For example, we can take our transcriptome-based assay and we can very specifically look at reactivation of HHVs in some cell therapies. That would be a concern, and determine whether it's coming from a donor material or if it's actually coming from the patient. So again, we are very active in this space to try to push these assays in front of the agencies so that they do really fundamentally understand what's going on with these assays and how they're used. Another area that we look into or we provide support in is we manage a type or maintain a type 5 biologics master file. If you're not familiar with a type 5, it's basically a repository of information. So we use it as a repository of information specific to our validation strategies for our different assays. So all of the supporting protocols and plans and reports and SOPs and any other supporting document, whether it's from the wet lab bench or is from the bioinformatics portion of our assays, they're all included there for easy reference, in particular by the FDA. So this is an FDA specific master file. We know that there are others, for example, Canadian ones. We're not quite there yet for those, but we definitely have one in place for the FDA. And we can gauge their understanding and their expectations by feedback we get both directly from the FDA as well as through our clients with very specific agency responses. And these are typically they tend to happen on Fridays at 5. We'll get I need a rapid response to, and here's the short list of questions from the agencies. And they tend to fall in a couple different categories right now that we're seeing. We see that the agencies understand what the technology is. Um, they understand the fundamentals, but when they dive deeper, they get pretty confused. And we're trying to work through with them to make sure that that does not continue, that they do fundamentally understand what's going on with these. Um, we see very specific questions that try to get us to push our limits of detection, provide more details about specificity determinations and sensitivity determinations and so forth, and really make sure that we meet the ICH Q2 or sorry, RCH, yeah, Q2 requirements in terms of validation for precision and robustness and so forth for the assays, and making them the most robust as possible to meet current expectations. And in fact, we've issued a number of letters of authorization, and we're seeing a lot of feedback based on those. And the feedback has been very good in terms of helping us further develop our assays to make sure that they stay current with new guidance documents that we expect to be around for about another 20 years. So with that, I'm going to end there. I realize I went through that pretty quickly, but I wanted to make sure we left sufficient time for questions. So I will then hand it back over to you, Jan. I would just thank the audience first for hanging in through my rapid my rapid presentation. Back to you, Jan.
Thank you, Colette. That was very interesting. Please do put questions in the Q&A if you have them. But no, that was really interesting. Maybe while we wait for some questions, I just had a couple that came to me while you were talking. I mean, in terms of you talk about the expert virologist, at the final stage, I guess, after the data analysis. I mean, do you see something like AI taking the place of that person at some
You know,
point? Are you doing
yeah,
AI stuff at the moment on that?
so we're always looking into things like AI and machine learning.
Mm,
And we have some internal initiatives for that as well. We have to start training on very different aspects of it. So, for example, not every
mm,
cell line is a CHO. So your data banks to start doing training can be pretty broad right now in terms of insect cell lines versus human cell lines versus so forth. We are trying to automate a lot of the processes. When you're talking
mm,
about a billion data points that you have to evaluate, nobody wants to do that by eye. So we have to automate a lot of that to simplify and not lose a lot of the context that we see. So yeah, I totally see AI as being a feature, at least to help the experts further condense the data down. At the end of the day, as the recipient of that data, you want a quick paragraph that says, I saw this and this is why I think it's significant and why you should be maybe it is of concern for you. What you don't want is a list of a million data points. So
mm,
yeah, absolutely. This is definitely an area that we have to improve on. We as an industry need to improve on. And the other one is just also turnaround time. I mean, the larger the data sets that you get, the longer it takes to process because you're
mm,
looking at an individual read by individual read and doing every bit of the analysis on that individual read, and then you move on to the next one. As these data sizes get larger, and we want to increase data sizes to some extent because it increases sensitivity, right? So you can start detecting the rarer and rarer molecule in there if it's an infection. But that also creates an impact on how quickly we can do data analysis. And we're very aware that our clients are asking for inline at line analysis and result, right? Real-time results. Right
mm.
now, we can do for our transcriptome assay 2-week turnaround time for a lot of our clients. They're like, can you do it faster? There's a fixed time in lab. We can certainly work on the bioinformatics portion of it. So yeah, great question.
And I guess just one other one, if while we're waiting to see if there's any other questions, just in terms of adoption, I know you offer this service for people, but there's a lot of components to think about in terms of adopting this technology, both
Right,
technologically, skills, capex, and
right,
bioinformatics, all that kind of thing. So, and also for regulators to understand as well, because sometimes they don't want just a black box, do they? And they don't want the answer, they need
right.
to know what's gone on as well. So how do you deal with that with your
So
customers
there
in
are
terms
now
of adoption?
exactly. So there are now roadmaps in place. So one of the regulatory guidance documents that I talked about, the European Pharmacopoeia, and I can probably see if I can go backwards here. Yeah. So 2641 is essentially now a
Mhm.
roadmap for how to, if you're going to set this up yourself, and we
Okay.
know that there's a few, commercially available entities that provide this testing service, but we're also aware that a lot of people are, a lot of industry is taking the technology in-house if they can. It's not the least expensive technology instrumentation is still on the higher end, and as well as setting up data analytics environments for processing and data storage. However, from a roadmap of how to do it, it's starting to become more and more standardized, and documents like 2641 do detail a lot of the requirements or the expectations, I should say, for that
Mm.
to enable it to happen. And we're trying to make it again through the consortias like CAACB and AVTIG and others, which are even nimble to some extent. Um, these are areas in which the industry can come together and say, okay, we recognize we're probably the primary players in this, but there's also going to be secondary players and those who are just now learning that they all need to get into this space and how can we help them? And that's where these roadmaps have really come from, is to say, okay, you guys have figured it out. Now let's share that information. Same thing with the agencies. We figured out how to present to EMA and FDA primarily, but then some of the other agencies, particularly in APAC, which tend to piggyback on, for example, the European guidance documents and that kind of decision making. We want to make it as seamless as possible so that they can take advantage of what's already out there and not have to start from scratch. So,
Yeah.
but good question.
Sure. Okay.
Okay.
Do we have any questions, Colleen? We have one, I think. Yes. So one question from Harsh is, for adventitious agent testing, which platform has more advantage, Illumina or Oxford Nanopore? Any comparative study done at PathoQuest for both platforms?
So again, the difference in the platforms between Illumina and Oxford Nanopore is in the length, is both length of the reads that you get as well as the depth of the data set that you get. So Illumina is significantly greater depth of data sets, even on the simpler platforms, than Oxford Nanopore. However, you can get different types of information out of that. So for a short read platform, that's essentially, you want to look in an ocean for the rare fish, not in a lake. With Oxford Nanopore, it's a little bit of the opposite. You're looking more in a lake, but you have more sequence read to give context as to something is intact or not. So there are advantages and disadvantages to both platforms. Both platforms have a place in adventitious agent detection. We primarily use Illumina now, but we are looking into Oxford Nanopore platforms because you can do other things like direct RNA sequencing that are not possible right now with Illumina-based sequencing. So we typically use the Oxford Nanopore platform for non-adventitious agent testing for things where you need long read context, like looking at replication competency, looking at genomic integrity of sequences. But there is a place for that as well if you want to say, well, I detect fragments of BVDV, but do I detect full length molecules of it? And that does make a difference. So we're trying to weigh that. I don't think there's necessarily I think both can be used equally well. I think what's used primarily in the field right now is Illumina-based sequencing because you can dive deeper and get more sensitivity. So if you have a very low level infection, you'll see it probably quicker on an Illumina-based system than you might see on an Oxford Nanopore. But that's current state. That could easily change. Oh, I think you're on mute, Jan.
Oh, apologies. There's one more question here. Is it true that NGS-based detection method is more sensitive than cell-based method? And how do we distinguish between functional and non-functional adventitious virus?
Okay, so if we're taking, again, the consideration is here is cell-based NGS, which is our transcriptome assay. If you think about in your in vitro test system, you have to generate enough infection for the cell to start showing some sort of cytopathic effect, right? Um, and that could be a significant level of infection, so over a significant period of time. Whereas in NGS, what we're looking for is the identification of those viral sequences. Now, there is the question of how do you determine or distinguish functional versus non-functional. We are looking very specifically at RNA. We are looking very specifically at replicating virus, transcriptionally active viruses, which is by looking at the RNA. So by default, there is a bit of extrapolation, but those are essentially the ground rules that we use to determine whether something is replicating or not, or functional or not, in a transcriptome-based assay. So it is taking molecular-based data, applying these other rules to it to make a biological determination as to whether something appears to be functional. We're also looking at things like you want to make sure you're seeing, we do, actually, I should take a step back. When we do our analysis, we do a protein-based analysis. We're doing nucleotide sequencing, but when we do our analysis, we're focusing more on the proteins. We do reading frame translation, all 6 frames, in order to determine whether or not something is actually going to make a functional protein and therefore be conducive to replication. So again, that kind of ground rule also establishes whether we're looking at something that's replicating or not by taking that approach. So great question. And I see someone has their hand up.
Yeah, I just saw that as well. Comfort, is it? Could you maybe ask your question? Come off. Colleen, can they come off mute? Yeah.
Right. Thank you so much. Very excellent presentation, very well aligned with our initiative at our lab, because we're trying to move to the use of next generation sequencing in the safety testing as a priority of going for the animal tests. Yeah, during the presentation, there was a highlight concerning the turnaround time. And this had been indicated as 2 to 5 weeks for next generation sequencing, which was indicated as being shorter than the 20-day assay. I got a bit confused there because I thought
I'll clarify it. Yep.
Okay.
So I'll clarify that. So yes, so we have what we call our standard assay, and then we have our, let me go back to the slide for that transcriptome one. Sorry. There we go here. So you're referring to this 2 to 5 weeks here. So our standard assay, one where we're not, you have to recognize currently NGS is still part of a package of assays in which there's always a longer assay in there. So from a standard turnaround point of view, we're probably in the 4 to 5 weeks. We're not rushing. We're just, we're applying our standard assay to it. However, from the shortest that this assay can be conducted right now is probably in the 10 to 14 day range. Again, you have a fixed amount of time for this entire laboratory process. And then really the driver of the full time is based on how long it takes to do data analysis. And the reason for that is the more it sees, the more we have to do secondary analysis, the longer it can take. But yes, this whole assay can be condensed into 2 weeks, which is certainly faster than a 28-day assay. Remember, 28-day assay is starting the assay. It doesn't include all of your upfront and your reporting. Our 2 weeks includes everything from study init to study complete, report issue. So again, that is the context that we use. We have a lot of clients who don't need a 2-week assay. We have, and it can be challenging to get these in queue to ensure 2-week assay, which is why we do have more of a standard 4 to 5 week assay turnaround time. But ultimately, this is inclusive of kickoff, study initiation, as well as reporting. And one thing I just wanted to add, and I forgot to mention this, from the reporting perspective, from a GMP perspective, we do not just provide a C of A that says pass, fail, conforms, or not. The agency expectation these days, particularly the FDA, and I've seen this evolve over the years, particularly through very specific conversations with the FDA, is they expect to see how well your sample does throughout this entire process. So they expect to see data related to these in-process control points. So what we do, is we provide a a final report in addition to the C of A that details exactly what each of these steps are for, what we followed, the methodology we followed, what criteria we used to say whether your sample met it or not. So either acceptance criteria, system suitability, testing criteria, and how well your sample did. And we provide this in addition to the virology expert report with a C of A. So you get a very comprehensive package out the door. And again, we can do that within a 2-week time frame under our rush assay conditions. Hopefully that clarified things for you.
Yes. I was asking it in the perspective. We recently had a session with one of the providers, Illumina to be specific, and they have this new platform. One of their latest, and they're saying with this we have shorter runs. So I was actually hoping that it could even be shorter, maybe a week. They were claiming it's faster, that the chemistry is better, and then they have a series of pipelines, standard pipelines, but probably would have to build one in-house to ensure that it's compliant with the regulatory requirements. So as just hoping it could even be shorter, maybe a week. Is that something you think is feasible, or it's an overstretch?
No. I think the technologies are moving closer to that space of getting to be shorter. You know, what we really need is real-time analysis of the data as it's streaming off of the instrument is the most ideal way to do it. However, not every instrumentation can do that right now. With some of the newer the newer Illumina instruments, I should say, they're changing their chemistries. They're more stable. They're room temperature, so you don't have shipping considerations, storage considerations. They can be run a little bit faster than you do now. They generate more data. So yes, the service providers, or sorry, the instrument providers and the chemistry providers are making some advancements. We get a little bit stuck sometimes because we make things GMP, which means we're fixed. We have to stay there for a little bit until we can do our proper revalidation of new assays and kits. But yes, the industry, at least the equipment sometimes moves faster than we can. So some of our platforms are still RUO, but yes, they are moving much faster, and we as an industry just need to keep up with that. So great question. So thank you.
Thank you. Thank you. Thank you very much.
Thank you for the question. We've got one more. Maybe we can squeeze this one in before the end, Colette. On similar lines, I guess. It's what QCs use to qualify a valid run?
Right.
How are the reagents defined, and where are they coming from?
So the controls are, again, they're assay specific controls. So we do have some in-house controls that we develop and we characterize ourselves. And then we use other ones like the WHO collection of viral standards. So this is a collection of 7 well-characterized viruses. They have a lot of characterization in terms of titer, genome copies. I mean, you name it, TCID 50s and so forth. That material is publicly available from the BEI. So again, it was initiated by Arifa Khan at the FDA. So there's actually 2 collections of those virus standards. The original one had 5. This one now has 7, plus a couple hangers on because 2 of the stocks actually have contaminants in it, known contaminants that we can use as extra trackers. So those we do use information available on the C of A's from those materials. Other materials that we use for our controls, again, they're just in-house materials that we do our own characterization on. For example, Mercy 5 cells, we have to characterize those. We use B95-8 Ramos, or we use Raji Ramos mixtures here, which are well characterized for that. This synthetic RNA control sequence is commercially available, and it's traceable. It's a well characterized stock for that. In terms of the QC, these other steps down here, these are just features of the specific module. So for example, after RNA extraction, we want to make sure we have sufficient RNA. So we use concentrations, we use purity assessments, and we use size assessments or intactness. So for example, a RIN number from a bioanalyzer trace would be, we set minimums for that. Library preparation, we do something very similar. We need a minimum amount of material that we can use that needed for sequencing, but we also want to check the profile of the library to ensure it meets the requirements for the particular system that we're using, that it has the right profile for that. Our sequencing, we have minimum quality scores, we have minimum data outputs that we need before we can even go into data analysis. From an assay QC perspective, we use detection of certain controls at certain levels with certain features. So for example, what's the coverage, what's the depth, how many reads do we see, how many reads do we not see, as well as other performance indicators of our pipelines before we go through this process here. And then everything down here, once you get to the virologist expert review, this is more, I'll call it subjective right now, which is where AI would really come in handy because you take the subjectivity out of it. But, you know, the controls here aren't really controls per se, but they do evaluate the controls from the entire performance of the run and the sample to say, I'm looking at good data and I can make conclusions on it, or I can't analyze this data because it failed a specific criteria at a step there. So great question. And I think we've coming at the top of the hour here. So
we are. I'll just thank you very much for that, Colette, and thank you everybody for your interest and questions. I've just got a couple of slides just to close out the session. So hopefully this just okay, so some closing remarks. So yes, thank you again, Colette. Really appreciate your time with this. In terms of AFSA and Humane World for Animals, we are continuing our project. It's funded by the Gates Foundation, and we have another 3 years funding to support this. So if anybody is interested in knowing more about the AFSA collaboration, please do not hesitate to contact myself or Colleen, who's also on this call, and we can discuss that further with you. I just wanted to remind everybody and give a save the date. We have a conference coming up, a global conference on animal testing replacement for vaccines with IABS, and that takes place in Bangkok on December the 2nd to the 4th. You can get all the information from our website and from the link here, and we're open for online registration now as well. So it'd be great to see people there to discuss more about the animal replacement testing of vaccines. So finally, I just want to say thank you to everybody for your attendance. This webinar will be available online, as I've mentioned, and a very special thanks to Colette and PathoQuest for presenting today. And I hope you all have a lovely day. Thank you very much.